Welcome everyone to exambees tutorial. In this video, we are going to discuss on a very important topic called photosynthesis. It is hard to overemphasize the importance of this process, which cleans the atmospheric carbon dioxide and convert it to life-supporting oxygen and food, ultimately essential to sustain life on the planet. We will unfold more facts about this very common yet important process as we progress in this video. Photosynthesis is composed of two words, photo which is light and synthesis means manufacture. So this is the process by which chlorophyll in plants use carbon dioxide and water to synthesize carbohydrate or glucose in the presence of sunlight. And the entire process needs 6 molecules of carbon dioxide and 12 molecules of water to form 1 molecule of glucose. 6 molecules of water and 6 molecules of oxygen are liberated as byproduct. And this extremely complicated process flawlessly takes place inside chloroplast, present only in the plant cells. But where these chloroplasts are located? If we observe a cross-section of leaf under a microscope, then we will see mesophyll tissue containing upper and lower epidermis. Just below the epidermis, there are compactly arranged columnar palisade parenchyma, and these cells contain chloroplast, which is the site of photosynthesis. Here, xylem present helps in bringing water to photosynthetic site. On the other hand, phloem takes part in transporting manufactured glucose to other parts of the plant body. And carbon dioxide needed and oxygen liberated in this process enters and exits through stomata. So does it mean that photosynthesis takes place only in a leaf? The answer is certainly no. It can take place in any green parts having chloroplast or photosynthetic pigments. Let us see how does the main site of photosynthesis looks like, that is the structure of chloroplast. These are double membrane structures having mainly two parts. Number one is a disc-shaped structure called thylakoid, which is the site of light-dependent phase. And second part is the fluid part called stroma, which is the site of light-independent phase. Chloroplastic DNA, ribosome, starch and lipid droplets along with thylakoid are embedded here, that is in the stroma. Whereas photosynthetic pigments like chlorophyll, carotenoid, xanthophyll are present within the thylakoid membrane. And stacks of thylakoid one over the other forms granum, which are interconnected by thread-like structures called lamella. Now we will unveil the true interesting part of our topic, that is the mechanism of photosynthesis. It occurs mainly in two phases. The first one is the light-dependent phase and the second one is the light-independent phase, which is also called Kelvin cycle. First, let us put some light on the light-dependent process. As I mentioned that photosynthetic pigments are present in thylakoid, so knowing detail of it would make our understanding much easier. Thylakoid is a double membrane structure containing an inner lumen, and the membranous part is composed of protein, lipid bilayer and protein, and photosynthetic pigments are embedded here. Pigments present here form light harvesting centers called photosystems. Two photosystems are photosystem 1 and photosystem 2. When sunlight strikes the surface of a leaf, chlorophyll molecules and other photosynthetic pigments are able to capture light but of certain wavelength. This means all 7 components of white lights are not equally absorbed. Chlorophyll A absorbs only violet, blue and red light of wavelength 430 nm and 620 nm. Chlorophyll B absorbs blue and red light of 453 nm and 640 nm. And xanthophyll, carotenoids, other photosynthetic pigment also absorb some light, but the maximum rate of absorption is shown by chlorophyll A, and the rest of the pigments are thus called accessory photosynthetic pigments. There are also other transmembrane proteins across the thylakoid membrane, which are electron transport system and ATP generating protein called ATPase. Mechanism of light dependent reaction has many intermediate steps. In this video, I have tried to give you an overall idea. You can visit our website for more details. First step of this entire critical but interesting process is light absorption. This process starts with the photosystems present in the thylakoid membrane. Each photosystem contains 300 to 400 chlorophyll molecules, proteins and other pigments. And there are two types of photosystem, PS1 and PS2. PS2 comes first in the electron path, but it is named as PS2 because it was discovered later. When sunlight strikes the chlorophyll molecule, then it absorbs photon and gets raised to an excited state. That is, one of its electron is boosted to higher energy orbital. And this energy is passed inward from pigment to pigment until it reaches the central part of photosystem 2 called reaction center. The reaction center contains a unique pair of chlorophyll called special pair. Here the electron is boosted high enough to get lost by a wavelength of 680 nanometer. And the lost electron is accepted by a primary electron acceptor called pheophytin. At this stage, the chlorophyll present at PS2 has a deficit of 1 electron as it has lost its electron and is positively charged. This deficit is fulfilled by getting 1 electron from splitting up of water, a process called photolysis of water. So now we can see that photolysis of water is very important, and this is carried out by manganese ion present in PS2. In the process, oxygen is liberated here as byproduct which escapes through stomata, and 4 hydrogen ions released into lumen increases the proton concentration of the lumen. Note this point here. We will see what happened to these protons a little later. Electron after leaving PS2 system is now transferred through electron transfer system comprising first plastoquinine, then cytochrome complex, and finally plastocyanin. When electrons pass from higher level to lower level, it releases some energy which pumps some hydrogen ions from stroma of the chloroplast to thylakoid lumen. This results in development of a proton gradient across thylakoid membrane. At this point, another important reaction takes place, photophosphorylation or ATP synthesis. The protons formed inside lumen would now tend to move from region of high concentration to low concentration, that is from thylakoid lumen to stroma, by a process called chemiosmosis. In doing so, a lot of energy is released which will be used in synthesis of ATP. This process is facilitated by ATP synthase or ATPase. This process is called non-cyclic photophosphorylation. In this process, ATP is produced by binding of 1-phosphate ion with adenosine diphosphate or ADP. So ATP synthesis is the third intermediate process and is very important as well. Now let us go back to the point and see what has happened to the electron leaving ETS of PS2. As the electron moves through this electron transfer system of PS2, it goes from higher to lower energy level and needs to be re-energized. This is done by absorption of 1 photon just before entering PS1 complex or PS1 700. The charged electron now travels upward through reaction center of PS1. Here other proteins of electron transfer system of PS1 takes their action, and in this series electron first passes through ferredoxin and then to an enzyme called NADP reductase. The enzyme NADP reductase helps in synthesis of NADPH by transferring 1 electron to NADP+, which is the 4th intermediate step, that is synthesis of NADPH. NADPH travels to stroma of chloroplast for further synthesis of glucose from carbon dioxide. Sometimes electrons released from PS1 is not taken up by any electron acceptor. Instead, the energy is used to produce ATP from ADP. And this process of formation of ATP is called cyclic photophosphorylation. This was an overall idea of light-dependent phase of photosynthesis. The products formed ATP and NADP will be used up in the light-independent phase. So let us now peep into stroma of what happens in the light-independent phase. This takes place in stroma of chloroplast and does not need sunlight directly. In this process, carbon dioxide entering through stomata changes to bound sugar with the help of ATP and NADPH formed in the light phase. The entire process occurs in 3 intermediate steps. Number 1 is the carbon fixation or carboxylation. Here carbon dioxide formed from light phase reacts with RuBP to form 6 carbon compound, which is very unstable, so it splits into 2 molecules of phosphoglyceric acid or PGA, which are 3 carbon compounds. This enzyme needed for this reaction is Rubisco or RuBP carboxylase. I have shown here that carbon has small red circles for better understanding. The next phase is reduction phase. In this phase, ATP and NADPH produced during light phase show their function. 3 PGA formed in carbon fixation process converts into 3 carbon sugar, and this process occurs into 2 intermediate process. At first, PGA, that is 3-phosphoglyceric acid, it combines with ATP to form 1,3-biphosphate glyceric acid and ADP. And this 1,3-biphosphate glyceric acid formed here will now combine with NADPH to form glyceraldehyde 3-phosphate or G3P. So here total 6 molecules of G3P are produced. Now the next phase is the regeneration phase. Only one molecule of G3P will be now utilized to make glucose, while rest of the G3P molecule will recycle to generate RUBP. And this entire process is explained by Kelvin, and so it is also known as Kelvin cycle. So this is all about photosynthesis. I know this is very complicated process, but a schematic diagram will help you to keep it in your memory easily. Don't forget to try our quiz at the end, and see you in our next video. Till then, happy learning!